Hello and welcome. Bonjour, bienvenue. I am Matthew Scotch. I am a professor at Arizona State University in bioinformatics, and I'd like to welcome you to our poster. This is collaborative work between Arizona State University and the National Library of Medicine's Intramural Research Program and the National Institutes of Health. The work is on wastewater surveillance of SARS-CoV-2 in the United States, and if you're interested in reading the paper on this that's been published in Science of the Total Environment, the citation is under the author's name and the QR code is at the bottom that you can take a picture on your smartphone. So here we describe the work on a near real-time, non-invasive, wastewater-based epidemiologic protocol that includes high-throughput sequencing and bioinformatics. The advantages here, and you probably read many papers during the pandemic on this, that it can be non-invasive in the sense that we're not requiring individual patients to get tested, and it can be done in near real time if done properly and correctly to give you a snapshot of what's going on in a specific population. It can certainly be extended to other viruses and has been as well, so it's not just relegated to the SARS COVID-19 pandemic. Because it's from wastewater and it represents a collection of samples from a population, you can't do traditional things like phylogenetics, for example, of individual isolates because you're not sampling directly from individual patients. So there are certainly challenges on the bioinformatics side, and some of which we try to address in this work through the development of new tools. So what we did is, and you can see the graphical abstract here, is we sampled from 39 sites across 9 states and 18 cities in the United States. We did this from July 21 to June 2022. And we also developed a few bioinformatics tools that certainly you are welcome to use. If you look at our full paper, we have the links there for the code and the tools. One is COVID-DIST, which is based on ordination analysis to measure the dissimilarity or distance between samples. So allows us to do principal coordinate analysis, which I'll describe in figures 3 and 4. And then we develop our pipeline for processing and analyzing the short reads from Illumina that we get from our wastewater samples. And that allows us to identify the single nucleotide variants, which can then be used in downstream analysis. So here's a look at our graphical abstract that includes sampling, processing, and of course our analysis. A little snapshot in figure 2 of what we're doing using our pipeline, our input files, our FASTQ short reads, Illumina reads, as well as SARS-CoV-2 reference files, as well as our primer files. We do alignment via BWA, we do primer trimming via IVAR, we identify primer bias via low-freq and IVAR, and then remove bias reads through IVAR, then finally try to call variants using low-freq and SNP EFF as well. So our outputs in terms of our analysis are variant call files and depth files, and then we're analyzing these through 2 main tools. One is our COVID-DIST tool that we developed, and then the other is Freya, which is used for lineage abundance. Just a little bit here, directing you right to figure 5. Just an overview of the sequencing data. We show here the breadth of coverage and the correlation with, on the x-axis with depth. Looking, so you can see there's some positive correlation there. And then that we're not showing you is we also have in other panels the time as well. So this is all by location. So a similar panel by time. And that, as we saw, that the quality of sequencing data was consistent among samples when looking at both location and time. And then finally in B, we're looking at the relationship between breadth of coverage and the CT values from one of our real-time PCR target sites. And then you can see there's not necessarily a statistical correlation here, but it's fairly apparent that there's not a clear correlation between the two parameters, suggesting that the result of a CT value is not necessarily going to suggest any sort of depth, breadth, and coverage. And then, I forgot to mention the breadth of coverage threshold for our study was 50% across the genome, so we removed any other sequences that didn't necessarily hit those thresholds. So looking at our results, figure 3 is a PCOA plot of our different samples with pangolin lineages of both Omicron and Delta. So the gray triangles are Omicron, and then the X's are Delta. And as you can see, there's clear temporal shift between Delta and Omicron, and even among the different clades. So 21J and 21I for Delta, and then 21K, and then 21L for Omicron. So as you can see with the July 21 samples to early November 21, and there's the legend that has the different colors for the different months, you can see that Delta significantly separated from Omicron along principal coordinate 1. And then you can see that Omicron emerged in early December 21 and superseded Delta lineages by late December 21 or early January 2022 at most locations. This shift once again mirrored what has been observed in the clinical data, as you can see with the triangles. Then the shift, and this includes BA1 derived lineages, which were more abundant in the initial Omicron wave of infection and were then later displaced by BA2. And once again, these results are consistent with data from our clinical samples. Looking at individual breakdowns of PCOA by state, our ordination analysis here suggests actually differences with clinical data. I know it's hard to read, at least here. If you see come up close to the poster in person, you'll be able to see it a little bit better. So by looking at the local spread within the state, we actually were able to unravel differences in wastewater between clinical samples. So we saw temporal shifts to Delta and Omicron that vary by state. So the shifts occurred in December for New Jersey, New Mexico, Washington, Georgia, and Illinois. And then for Arizona, Kentucky, Oregon, and Texas, this occurred in January of 2022. We see that Omicron BA1 lineages are displaced by BA2 lineages around March in the clinical data. And in our wastewater, we see it, for example, in Arizona in February, and then in April for Georgia, Illinois, New Jersey, Oregon, and Washington. Finally, we look at figure 6, which is our use of Freya for identifying VOC-defining mutations. I know it's super hard to read, so apologize for that. But we can see the top x-axis here are the different sites, and then the breakdown by months. On the y-axis are single nucleotide variant defining specific lineages, including those found in Delta and Omicron. And so if you go to the bottom, you can see there's some variation between what happened in our Omicron and then what was seen in other samples as well. So Omicron defining single nucleotide versions were earlier than December 2021, which is when most people found out about Omicron. So here, for example, I know it's hard to read, but you have the arrow and the circle showing, for example, New Jersey showing lineages specific to Omicron or variants specific to Omicron lineages coming in very early, for example, in September. And then there were also Delta lineages that were missed completely by clinical data. And then we also observed the presence of Delta and Omicron variants. Figure 7, you're welcome to check out as well. That's our analysis of lineage abundance across the different states. And we even show that the presence of recombinant lineages in these green, and that actually had impact on the eventual displacement of Delta to Omicron. So again, thank you very much for visiting our poster. Hope you enjoyed it. Encourage you to read our paper in full, and just want to finish up with the discussion and say that there's no doubt that wastewater surveillance can provide important data on viral diversity of circulating variants of SARS-CoV-2, and that lineage abundance certainly showed the value of these neighborhood level implementations that we saw both in the PCOA as well as with FRIA. There were things that were missed by clinical sampling, so in order to get a more robust snapshot of what's going on in the population, it's certainly important to couple traditional surveillance with wastewater-based epidemiology. And there's also the potential to identify things like recombinant lineages. So thanks very much for funding National Library of Medicine, National Institutes of Health through our RADXRAD initiative, and then the NLM's Intramural Research Program and the J.M. Kaplan Fund. Also wanted to point out a declaration of competing interest for Dr. Halden and Dr. Driver, who are co-founders of Aquavitas. Thank you very much. Feel free to email me at matthew.scotch@asu.edu, and hope that you enjoy the rest of the conference. Okay, bye-bye. Au revoir.